Biotechnology is the use of biological agents, living or parts of living organisms, for technological advancement. Aspects of biotechnology can be implemented to maintain a living stock of diversity, remove undesirable or disease-ridden traits, isolate metabolic products for therapy, increase yield, decrease costs, the ability for us to advance in biotechnology, the ability for us to apply these findings and facets to the wide variety of biological fields really depends upon two key things. One, the technology at hand, and two, our knowledge about the available living organisms and their utility. The first technique is DNA extraction. The goal here is to isolate and remove the genetic material from cells. To do so, you must break the cells to access the nucleus, break open the double-membraned nucleus, remove the proteins upon which the DNA is coiled, and then precipitate the DNA out of solution. The process of PCR involves manipulating a DNA sample using temperature to replace the roles of many enzymes. Heating slightly denatures the chromosomes, allowing the DNA to unwind. By cooling it, the DNA strand can be stabilized and the primers can bind. And then a slight temperature shift will stimulate the polymerase to bind and perpetuate elongation, because that last temperature shift is at its optimal temperature. The significance is that repeat cycles of this can be done on its own automatically and relatively quickly. You could get hundreds of copies in the span of a few hours. Once many copies of DNA are made, a likely logical step is to then sequence the DNA. Determine the unique sequence of A, T, C's, and G's, the unique genetic code. To do so, you have to conduct a specialized PCR using some nucleotides that are radioactively tagged. And so a tube for each nucleotide base is used, one for the A's, one for the T's, one for the C's, one for the G's. Each of these tubes will also get primers, the same DNA sample, as well as a working DNA polymerase. And within each tube, we'll see that heating and cooling and slight heating shift cycle repeat. And so the DNA will unwind and the primers will stabilize and the polymerase will start copying. But when it adds a tagged nucleotide, the additional bulk from the radioactive label will result in the polymerase falling off. And when it rebinds and copies again, as long as it's adding regular nucleotides, it will continue down the sequence. But once it adds a radioactively tagged one, it will fall off again. And the result is that you get these different size fragments. These fragments coincide to location and place of that particular nucleotide in the genetic sequence. And we can determine this by running it on a gel, which will use diffusion to organize these sample fragments by size. Big things are heavy, and so they don't travel far, but small fragments are light and they travel very far in a short span of time. And so when you have this gel, you can read from the bottom up to determine what's the first nucleotide in the sequence, second, third, fourth, all the way to the end. This type of gel looks at nucleotide polymorphisms in the sequence. Another process can compare fragment length polymorphisms, and this is looking within specific gene areas where there are palindromic sequences that are subjected to insertions and deletions. Using specialized enzymes to cut at these sequences will result in different size fragments within one or two particular genes, creating a unique banding pattern, which can allow us to look for similar patterns amongst closely related individuals or species. So in this particular example, we've got mom, her child, and three potential fathers. We can identify the bands that came from mom and then look to see which male can fill in those gaps. Similar process is done when we identify a criminal at the scene of a crime, or for immigration disputes, or identifying an unknown individual during times of war based upon a small DNA sample. Another technique that is up and coming is genetic cloning. Not to be confused with organismal cloning. Gene cloning is when we insert a gene of interest into a model organism. We could be adding a human gene for production of insulin into a bacteria because they're easier to grow and monitor. The process of this uses restriction enzymes, and that just means an enzyme that cuts the DNA at a particular place. And these cuts are at a diagonal, leaving sticky ends. So the DNA is much more likely to pick up surrounding genetic information to fill in the gap. And so you apply the gene of interest that you want, a little heat to stimulate the uptake and repair. Some of the bacteria or yeast will repair it perfectly without taking in the gene, but some will pick up the gene of interest. And those we would recognize as recombinant DNA. They've recombined an outside genetic source. In order to confirm who picked up the gene and who didn't, we typically add a tag to the gene of interest, either color or antibiotic resistance. With a color tag, those bacterial colonies will grow a different color, and so we know if you picked up the color gene, then you also picked up the insulin gene. Or if we looked at an antibiotic resistance gene, we would then grow a selective media culture, and so anything that grew had the resistance gene, and if they had the resistance gene, they also picked up our insulin gene as well. A karyotype is a visualization method to organize all the pairs of chromosomes within a cell and identify structural anomalies. While today's technology allows us to multicolor code and sort through artificial intelligence, in the past, this was all done through jump sustaining and a comparison of light versus dark banding patterns, and done by eye. The process involves extracting the chromosomes from a nucleus and then arranging the pairs first based upon size, with the tallest at the front and the smallest at the end, reserving the sex chromosomes for the very last. Should there be two chromosomes about the same size, as you might see in row 2, we then organize based upon the position of the centromere. The centromere holds the two sister chromatids together, and it can occur right in the middle, in the case of a metacentric centromere, or slightly towards the top, submetacentric, very near the top when it's acrocentric, and at the tippy top when it's telocentric. In the event that a chromosome is the same size and has a centromere in the same location, the third hierarchy is based upon the banding patterns, the proportion of light versus dark. By using karyotypes, we can recognize disruptions in chromosome number or structural anomalies. The breakage of a chromosome can lead to 4 types of changes in chromosome structure if not repaired correctly. The causes vary and can occur anytime, from radiation, chemical damage, ultraviolet light, viral infections, or simply just errors during synthesis. Most of these are repaired, but some are not caught or repaired properly, and this can result in chromosomal abnormalities. In chromosomal deletion, just a portion of the chromosome has been excised. During duplication of a chromosome, a segment has been duplicated, as its name suggests, lengthening the overall chromosome. This can be problematic because when expression occurs, you may now be expressing 2 or 3-fold the amount of those genes and their proteins. Inversions occur when the break results in flipping the entire sequence. And translocation is the only error that involves 2 different chromosomes, where one chromatid is broken and moved to a completely separate chromosome. Another cause for concern is non-disjunction. What if the chromosomes don't separate correctly during anaphase? This can lead to aneuploidy, an abnormal chromosome number. Aneuploidy describes a situation where one gamete receives 2 copies and another gamete receives no copy as intended. The outcome of non-disjunction depends upon which round of meiosis the sticky chromosomes occurred in. If it occurred in meiosis 1, then all daughter cells will be affected, either having too much or too little. But if the non-disjunction occurs in meiosis 2 to just one of the cells undergoing anaphase 2, you're looking at 2 normal daughter cells and potentially 1 cell with too much and 1 cell with too little. What makes non-disjunction more or less likely? Maternal age over 35, a previous child born with an aneuploidy, a history with previous births resulting in aneuploidy, being a carrier of a genetic abnormality, either a translocation or inversion, or being an aneuploid yourself. Risk factors that could increase your likelihood of passing an aneuploid disorder typically result from an advanced maternal age. There are a number of aneuploid diseases: monosomy, where an individual only gets 1 copy of a chromosome; trisomy, where they get 3 copies of a chromosome; and polyploidy, which results in 3 complete sets of the genome. There is only one viable monosomy known to existence, monosomy X, also known as Turner syndrome. This is the result of non-disjunction of sex chromosomes. Either the XX or XY, mom's egg or dad's sperm, failed to separate appropriately, and so one gamete got both sets of information while the other got zero. When that zero sex chromosome gamete fused with the other, the resulting offspring had only one sex chromosome. This is rare. With no Y, they are considered female, although they will have some physical anomalies, such as a short stature, broad chest, low hairline, webbed necks. Girls with Turner syndrome typically experience gonadal dysfunction, non-working ovaries, which will result in sterility. Any chromosome could be subject to non-disjunction and result in a trisomy inheritance pattern, but almost all will result in spontaneous abortion or miscarriage within the first few weeks of fetal development. The exceptions being trisomy 21, 18, and 13. Trisomy 21 gets its name because it is 3 chromosomes of the 21st chromosome. The layperson name for this disorder is Down syndrome. It exists in roughly 1 in every 700 births. Individuals with this inheritance will have physical growth delays, characteristic facial features, and mild to moderate intellectual disability. The average IQ of an adult with Down syndrome is the mental capacity of your typical 8 or 9-year-old child. Trisomy 18 is also known as Edwards syndrome, is the result of 3 copies of the 18th chromosome. This is rarer, but it's an interesting disorder because females born with this abnormality are more likely to survive infancy, whereas males with this abnormality are typically delivered as stillborns. Individuals with an extra 18th chromosome will typically inherit small heads and jaws, an intellectual disadvantage, heart defects, and they're typically born with a strange clenched fist with overlapping finger presentation. Our final trisomy that we'll discuss is trisomy 13, also known as Patau syndrome. This is really rare. It results in the failure of the forebrain to divide properly. That's holoprocephaly. This will result in intellectual disability and prominent motor dysfunction. It's also common with individuals of Patau syndrome to exhibit extra fingers. Polyploidy, again, is the result of non-disjunction of all the chromosomes. It is very rare in animals, but common in plants. And the result is a complete duplication of the genome. This is the result of non-disjunction in anaphase 1. The overall value of doing a human karyotype is the ability to not only look for structural anomalies, the occurrence of significant trisomy or monosomy disorders, but also detect gender. Here we can see the correct number of chromosome pairs. There's no additions or deletions, no trisomy or monosomy. And because at the very end, we can see a single X and Y, we know that this combination is a chromosomal biological male. Gene therapy, where we can move one gene. Could we move functional genes from one human to treat or prevent disease in someone else lacking that genetic instruction? There are 2 types of gene therapy. Know the difference between ex vivo and in vivo. Vivo means life, so ex vivo means outside of the body, outside of the living organism. The cells are removed, they're manipulated, and then they're re-put in. In vivo means all of this is happening within the body. The gene therapy is introduced to the person and then has to navigate the entire organism system to find the specific cells of interest, target those genes, and replace them. How can we achieve this? By using stem cells to repair damaged tissues, or using viruses because they are, at their core, genetic delivery mechanisms. Gene therapy is not the end-all be-all cure at this point in time. It's limited to somatic cells and disorders that are caused by a single gene. But that still opens the door to a number of diseases having treatment and potential curative options where before there were none. And ultimately, this is an alternative to a lifelong prescription of drugs or a very invasive surgery. As we continue to expand on genetic engineering, it helps us to understand the goal of this process. It's not to meddle or play God. It's to help organisms become better suited to the environment or human use in a shortened time frame. Genetic engineering has always been a process through selective breeding, interspecies crosses, mutagenesis, transgenesis, and new breeding techniques like CRISPR and RNAi. Some of these processes happen in the wild with or without human intervention, and they all have their own level of human influence. But often when we talk about genetic engineering, people get nervous. There's controversy over genetic modified organisms. People are worried that they may contribute to an increase in food-related allergies, antibiotic resistance, cancer. They're worried that there's a monopoly on these genes and this technology, and whether or not we can control the spread of good genes or bad genes. There's the argument that if these things naturally happen in the wild, why should we be meddling in them? But the pros outweigh the cons. In agriculture, we can improve the nutritional quality of food. We can make it easier for farmers to grow with less fertilizer and water requirements. We can increase crop yields. By relying less upon pesticides and herbicides, we can have large yields with minimal chemical intervention, which affects our water table. And in terms of safety, the FDA requires all genetically modified foods to meet the same requirements as anything else. And we've seen genetically modified technology help reduce the price of crops. The utilization of pesticides, which affect other wild plants and animals. Remember, genetic engineering isn't new. We previously called it selective breeding. We were engineering better plants. Native bananas are not edible because they have very, very large seeds. But by breeding and crossbreeding and breeding and selecting for bananas that had more fruit pulp versus seed, we have created the banana that you know today. Through selective breeding, the wild mustard plant has given rise to 6 different vegetables that we see in our grocery stores. Now, nature has done this on its own, but that takes thousands, sometimes millions of years. By selecting for the traits we want, humans can do it in 10 to 100. Genetic engineering moves 1 to 4 genes only. We know exactly what we're moving and where we're putting it. It's instantaneous. It's in the next generation or it's not. No intermediates, no hits and misses. We can check for it. And so genetic traits have been approved in close to 400 plants worldwide and about half of them in the United States. We've made canola that are herbicide tolerant. We've made papaya with disease resistance. We've made soybeans healthier by adding high oleic content and increased omega fatty 3s. Genetic engineering allows humans to respond to an environment that's changing at a much more rapid pace. All of this is possible because the genetic code is nearly universal. It is shared by the simplest bacteria to the most complex animals. And so A, T, Cs, and Gs, that order of a particular gene can be moved and transcribed and translated in any species, so long as the instruction is there. Here we see bioluminescence from a cyanobacteria transplanted to a plant and a pig, showing just how universal this instruction is.